Good evening, everyone. So, till yesterday, we have started with the genetics and we discussed about Mendel's laws of inheritance. So, I discussed about monohybrid cross and dihybrid cross and how we get genotypic and phenotypic ratios. So, I hope it's clear. Now, other than this monohybrid and dihybrid crosses, there are some other crosses as well. So, we will also discuss them. So other type of crosses are polyhybrid cross. So polyhybrid cross is nothing but a cross involving three or more gene pairs. So yesterday in the monohybrid cross, we have discussed only about a single character, that is height. And in dihybrid cross, we have discussed only about two characters, that is color and shape of the seed. Okay. So then in the polyhybrid, we can take multiple gene combinations, gene pairs, like 3, 4 or more. Okay. And we will draw a checkerboard, that is Punnett square. And accordingly, we can calculate the ratios, genotypic and phenotypic ratios. Then direct and reciprocal cross. So crosses are of two types based on the way they are crossing, that is direct cross and reciprocal cross. So a cross between two individuals is called as direct cross. Whereas when we change the order of the individual in crossing by reversing the male and female, it is called as reciprocal cross. So that means like in plants, like suppose capital T is there in the female part, that is egg cell, and small t, that is dwarf character we are selecting from the pollen. So that is direct cross. But if you are reversing, the dwarf is from the female and tall is from the male. Okay, we are reversing the characters. Then that is called as reciprocal cross. Then next is back cross and test cross. So back cross is nothing but mating of F1 generation, heterozygous F1 generation. So heterozygous means I told you yesterday what is homozygous and heterozygous. So homozygous is where the two copies of the gene, that is two alleles, are of same time, whether it is capital T or small t, for example. That is homozygous. Homozygous dominant, homozygous recessive. Heterozygous means one copy of each type of allele, that is capital T, small t. That is called as heterozygous. Okay. So then F1 generation heterozygous, that is capital T, small t, for example, it is crossed or mating with the homozygous parental generation individual. That means the capital T, small t may be crossed either with capital T parent or small t, small t. Okay. That is, we are crossing it at the back. We are going to the past generation. That is called back cross. Now, back cross can be of two types, out cross or test cross. When capital T, small t is crossed with the homozygous dominant parent, that is called outcross. Okay. And when, so what happens? Capital T, small t, it is crossing with the capital T. So capital T will definitely give only capital T, right? So homozygous dominant parent is giving only the gamete with capital T. Now this heterozygous F1 progeny, it gives capital T and small t. So that's why here we get combination of capital T and small t. That is one combination, capital T is one combination, capital T, small t is one combination. So we will get two combinations. Okay. Then in the test cross, this heterozygous the progeny is crossed with the recessive, homozygous recessive parent. That is called test cross. That is capital T, small t is crossed with the small t. So here you can see. Now capital T, it combines with the small t in all the individuals. Okay. Sorry, in the two individuals. And small t is combining with the other small t. So 1:1 ratio will get. If there are four individuals which are generated, two will be having heterozygous genotype and two will be having homozygous recessive genotype. Is it clear? And we'll have genotypic ratio and phenotypic ratio. I told you what is genotypic and phenotypic ratio. Now, what is a phenotype and genotype? So, irrespective of the genotype, suppose you see an individual. He is having good height, maybe 6 feet height. So then you actually don't know what is the gene. Okay, what is the combination of gene, and you don't know what kind combination of alleles and all that. Only character you can see height is he's tall, or he is dwarf, or she is tall, she is dwarf. So that is the character, physical appearance of an individual. So it is color or height, sex, etc. Okay, like in here, purple color flowers, white color flowers, like that. So they are called phenotype, irrespective of the genes they are having. Gene will be same, but what kind of combination of alleles it is having and all that you don't know. Okay, that is called as phenotype. So it refers to the visual characters like color, shape, stature, plant surface, etc. And individuals with a similar phenotype may be homozygous or heterozygous. So because homozygous dominant definitely give the dominant character, heterozygous also, although recessive allele is present, but the dominant allele will express itself, masking the expression of the recessive allele. So the person may be having either homozygous dominant or heterozygous genotypes. And for single gene with complete dominance, the phenotypic ratio will be 3:1. Okay, that is called as complete dominance. You have some exceptions. If it is there in your syllabus, we will discuss that also. So that means you don't have any confusion here. So completely that allele is dominant over the other allele. So in such case, usually the phenotypic ratio it will be 3:1. That means if there are four individuals, three will be tall, one will be dwarf, like that. And the phenotypic classes are always less than the genotypic classes. And genotype is nothing but the genetic constitution or the makeup. So that is, now you have to, when you check the genes, so whether they are having homozygous dominant or homozygous recessive or heterozygous genotype, so that combination of alleles is called as genotype. Okay, so it refers to the genetic constitution of an individual, and individuals with the homozygous genotypes will always be true. And for the single gene, the genotypic ratio will be 1:2:1. Okay, and the genotypic classes are always higher than the phenotypic classes. Okay, now the Mendel's laws of inheritance. So what he has done? So Mendel has given the foundation for genetics. So that is the basic principles of heredity. He conducted experiments on chickpea or garden pea, which is called Pisum sativum, in during 1856 to 1864, that is for 7 years. And he proposed three important laws of heredity. What are they? Law of dominance, law of segregation, and law of independent assortment. So what is law of dominance? So that is, one allele is dominant over the other. Okay, that is the one which expresses itself even when other allele is present. So that is nothing but the dominance. Okay, so he made monohybrid crosses, tall and short. All this we have discussed yesterday, so I'm skipping all those things. So it is nothing but that one homozygous condition. In homozygous condition, definitely whatever the allele is present, that will express. Okay, no doubt about that. Like if it is capital T, definitely the individual will be tall. If it is having small t, definitely the individual will be dwarf. So there is no confusion about that. In heterozygous condition, that is, if it is having capital T, small t, then which allele expresses itself even in the presence of other allele? So that one is nothing but the dominant allele. And that phenomena is called as dominance. Okay, this is called as law of dominance. Always one allele will be dominant over the other, mostly. But there is some exception where you can see codominance, where both the alleles also they equally express. Okay, so there are a few examples regarding that also. But majorly, some alleles are dominant over the other. Then law of segregation. So it is the first principle of heredity where there are two factors or two genes for each character. That is two alleles. Like capital T, small t. Okay. Then they segregate each other. Okay, they segregate this capital T. There are two copies of capital T, right? Now, when the gametes are formed, okay, there is no such case where both these capital T's goes into one gamete and other gamete is having no allele. There is no such condition. So these two alleles, capital T and capital T, must separate. Segregation means separate, and each gamete will be having this one of the alleles from the parent. That is called as segregation, law of segregation, and that will again pass on to the when after mating again that will combine with the other allele and again that will pass to the next generation. Okay. Then law of independent assortment. Here, this is a second law, second principle, where so different pairs of contrasting characters they assort independently of one another during the gamete formation. That means if you see yellow and round seeds and green and wrinkled seeds or any combination, you don't see like if there are two characters. Suppose in human beings, I am telling you example. Suppose a person who is tall is having straight hair and a person who is dwarf he is having curly hair. So do you expect that all tall people will have straight hair? No. And all people will have curly hair? No. There is other combination also possible. Even tall people with curly hair and short people with straight hair is also possible. That means these are two characters, type of hair and height. These two, they don't depend on each other, right? If tall height, if we is tall, then definitely straight hair should come is no. So these two are separating independently, assorting, coming, like inheriting to the next generation separately. So there is no association between the two different characters. So that is nothing but the law of independent assortment. So like you can get different combinations of characters. If the alleles for different characters are present on homologous pairs of chromosomes, they are governed by the phenotype of linkage. But sometimes, if they are present very closely, these two genes are present very closely, then there is a phenomena called linkage. So some genes may be in linkage that we will discuss. But otherwise, they are separately inherited. Okay. There's reasons for the Mendel's success. Okay. So although different scientists have performed different experiments, maybe before Mendel or after Mendel or whatever it is, still Mendel's laws were 100% successful. Even today, if you perform the same experiment with same strategy, you will get the same results. Now, even in the Punnett square, if you do calculations, you will get same ratio. Okay, so why he could get such kind of success? That is, first is proper maintenance of records. So this is very important. See, not only for you, although you are in UG level, but if you go to PG or PhD or even in the professional level also, like anywhere. So this is where if you want to continue in the research field or anywhere, like proper maintenance, systematic observation is important, and maintenance of records is also important for different generations. So that is what he has done first thing. Because we will write something on some paper and we'll forget where it is. Again, next time if we want to do, we don't find the papers. So it will be clumsy. Okay. But even if it is failure, even the experiment is failure. Okay, note down what is wrong in that. Okay, this combination has failed. Okay, fine. Maybe that can give a lead to the next time experimentation. Oh, you should not repeat such kind of formulation, right? So that is first thing. Second is a study of individual characters. So although he selected 7 characters, he did not started all at a time. Okay, he studied only one by one, only one character at a time. So this has given the clear idea. Okay, then choice of material. So he selected garden pea. Why? Because it is hermaphrodite. Male and female are on the same individual, self-fertilized. It has short duration crop because if it is taking longer times, so the experimentation also, you cannot generate multiple generations here. Okay, as it is having a short crop time, you can raise multiple generations and you can observe uh, like number of generations and how the inheritance is happening. Okay, and this is also very easy, like for maintaining of purity of material and raising more than one generation in an year. And the flowers of garden pea are also very easy for pollination. He has done some self pollination, like selective pollination. So everything that is easy, and you can even it doesn't require any much space. It is a garden pea. Even in the garden, even in the pot, also he could perform. So that is very important. Maintenance of purity. So here the parents or lines of hybridization are very pure. So he has studied individual characters and he could study them in a systematic way. Okay, without any contamination. Okay, then knowledge of shortfalls of earlier work. So what are the limitations? So what kind of failures the earlier workers have met with? Okay, so that he actually he got the knowledge about that, so that will give him that he gave a new direction and better planning of his work. Then mathematical background, he has a very good physics and mathematical background as he studied in University of Vienna. So that's why the calculation part everything was perfect. Okay, then proper choice of characters. So even the characters, he took only contrast characters. Not he took the characters which are of long range. Like suppose he has only like yellow or green, but not yellow, greenish yellow, bluish yellow, blue green yellow. No such kind of different combinations he did not find. Okay, only contrasting characters, whether it is yellow or green. That's it. Yes or no. That's it. Okay, no range here. So this gives a clear idea, clear picture. Then reasons for overlooking of Mendel's results. So Mendel pretended results of his 7 years presented, and he devoted work in the meetings of Natural History of Society of Brunn in 1865 in 2 papers. So his papers were experiments in plants in the annual proceedings of the society in 1866 in German language, and they were distributed in several libraries. That time it was only in libraries, but now you can find all the research articles even in the websites. but that time only in the library you can find. That is Europe and America. And however, Mendel's work remained overlooked for 34 years. Maybe because there was no knowledge about genetic material or DNA structure, or whatever may be the reason, or only few scientists were there at the time. So maybe whatever may be the reason, his papers were not studied or not carried forwarded by many people. So he died in 1884. Okay, after his death, so three different scientists, De Vries from Holland, Correns from Germany, and Schermark from Austria. So they independently, they have not in collaboration, and they were not colleagues or so. Independently, they worked, and then they could come to the same conclusion what Mendel has published. Okay, so that's why these are the important reasons for neglecting the Mendel's revolutionary findings. So after they again republished or reconfirmed the work, then by the time some amount of research has done, some progress was there. So maybe that is because people showed interest. And then the genetics field has developed, and he was named as father of genetics and all that. So why actually his work was went unnoticed? Maybe Mendel generalized his results based on his studies on garden pea. Later on, he worked on hawkweed on the advice of C.V. Nageli, and Mendel could not prove his results on this plant as an embryo is formed from the ovule without fertilization. This created doubt in the mind of scientists that the results Mendel got in his experiment in Pisum plant. So, although he got some results, as I told you, there will be definitely some exception. Even here, like codominance, then epistasis, such kind of terminologies were later developed. So, what Mendel did was he generalized the concept to every organism. In garden pea, okay, he got this kind of inheritance, but in others, the way may that the process may be changed, or there may be some exception, but he did not focused on that. He generalized the same thing on all the plants and all the organisms. Due to that, actually, in some other organism he could not prove the same results. So because of that, the scientists doubted his work. And since Mendel has got good background of mathematics, he explained his results with the help of mathematics. But scientists did not appreciate that approach. Okay. See, even for now, if biology student, you explain all the things in mathematics, some big theorem, even though you show the results, maybe it is correct, but you don't understand anything, right? Without some proper background knowledge, even if we like take up a lot of mathematical work and all that calculations and all these things, it looks like it is a theoretical part but not the practical one. Unless we do the practical work, it doesn't look like a biology part, right? So maybe because of that nobody showed interest on that. And Mendel could not support his findings through cytological investigation or cytological studies. Similarly, I told you, although he did on garden pea, unless we go to the wet lab, we perform the experiment and we are getting something which we can see with our eyes, so it is not possible to believe. So although he got some theoretical knowledge based on some mathematical works, it looks like a hypothetical theorems. But when he did experimentation, he some plants he could not prove the results. And that is because not his fault, but the cytological procedures were not well developed during that time. And after his failure to demonstrate the results of an hawkweed, he lost interest in research work. Okay, and he was a monk, I told you. So he dedicated to the monastery, and he did not give proper publicity to his work, and he kept quiet. But Mendel published his results in a rather obscure journal whose circulation was limited, and very few scientists of reputed journals have read it. Okay, so that's why even now, mostly researchers, they encourage to publish in a very good paper. Like there are Nature, Lancet, like that. So which is prestigious journals, and everybody reads those journals, and open access journals. Everybody will have the access. But he did only in small journal which nobody read. So obviously his work was not reached. Okay. So other than this, there are non-Mendelian inheritance. So the basic principles of heredity was given by Mendel. So there are some non-Mendelian inheritance also. They are called as Mendelian deviations, exceptions, and anomalies. They are like incomplete dominance, codominance, multiple alleles, linkage, lethal genes, gene interactions, pleiotropic gene effect, then polygenes, environmental effects, and cytoplasmic or maternal effects, which all is he did not find. Okay, what are these concepts? We'll see. First is incomplete dominance. So here in Mendel's experimentation, I told you one allele is completely dominant over the other. But here, one allele is, although it is dominant, but it is not completely dominant. Okay. For example, in 4 o'clock plant, Mirabilis jalapa, there are two types of flowers. One is red and other is white. So if it is according to Mendel's laws, either red or white should be dominant, and only that should come to the next generation. But when a cross between the red and white flowers was performed, they got pink color. It is intermediate in the F1 generation. So now the ratio also will change. So you got 1 is to 2 is to 1 instead of 3 is to 1. Okay. So that's why it is called incomplete dominance. Then codominance. Codominance, both alleles will express. The heterozygote, so both the dominant allele and recessive allele both will express at a time. The best example is AB blood group of human beings. So usually there are 4 different types of blood groups, common blood groups: A, B, AB, and O. So when A allele is expressed, A blood group, then B allele is expressed, B blood group. Then no allele is expressed, it will give O. When both A and B are expressed, then it will give AB blood group. It is a heterozygous exhibiting phenotype of both. Okay, this is called as codominance. Okay, then multiple alleles or allelomorphs. So the best example is coat color of rabbit and blood groups in man. So multiple alleles means, I told you, like alleles only 2 forms. One is dominant, other is recessive. That is what we have discussed yesterday. But there are multiple alleles also. In the coat color of rabbits, so usually the rabbits will have different shades of coat color. Coat, like hair, which is covering. So the coat color in rabbits exhibits 4 colors: agouti, chinchilla, Himalayan, and albino. Agouti is the normal coat color, where it is full color in which they have band hair. So here you can see agouti, which is black. Okay. So the portion nearest to the skin being succeeded by the yellow band, and finally a black tip, because it is dominant character. Okay, it is represented by capital C. Then chinchilla. Chinchilla is, it lacks yellow pigment, but it is having black and gray. So its hair appears like a silvery gray. It is called chinchilla. Then Himalayan, it looks like a bit white, except extremes are black. Okay, third one. Then albino, it is complete white. okay, including eyes. So when crosses are made between the individuals with different coat types, it revealed that agouti is dominant to all others and albino is recessive to all. Okay, so like that. So here you can see the hierarchy: agouti, then chinchilla, then Himalayan, then recessive one is albino. So this is how the genotype looks like. If it capital C is present, it will give agouti, that is black color. If both alleles are chinchilla, it will give chinchilla, that is gray color. If it is chinchilla along with Himalayan or chinchilla along with the other one is albino, it gives light gray only, because Himalayan is chinchilla is dominant over Himalayan. If Himalayan is present, either homozygous or in combination with the albino, it gives Himalayan albino. And albino is present only if both the alleles are albino. Is it clear? Then next is blood groups in man. So here the discovery of human blood groups was actually reported by Karl Landsteiner in 1900. So for his important discovery, he was awarded Nobel Prize in Medicine and Physiology. And this Landsteiner's work led to the establishment of 4 common blood groups: O, A, B, and OB. So AB. So here they have antigens and the antibodies are produced. Okay. And in the ABO blood groups of the factors, there are genes which determine the antigens and antibodies. And the antigen appearing in the offspring only present if at least one of the parent is having that. And the 2 antigens that were discovered by Landsteiner are A and B, and he could find like this a table. The person having O blood group is having genotype with both are recessive, no antigen. Okay. But antibody is anti-A, anti-B. Okay. Then A blood group, they are homozygous for A or heterozygous. So A antigen is there and anti-B antibody. For B blood group, antigen B and anti-A antibody. AB is both A and B antigens and no antibodies are produced. Okay. So here multiple alleles are present. Okay. Other than 2. Then linkage. So linkage is nothing but actually this is a deviation from the independent assortment of genes. So the cases of linkage was reported by Bateson and Punnett in P, Hutchinson in maize, and Morgan in Drosophila. So here when there is a linkage, they got the ratio 1:1:1:1. Okay. So that means the genes which are present very nearby, they inherit together. There is no independent assortment. Like the tall person with, suppose the gene for the height and hair type, that is tall person with a straight hair. So if the 2 genes are present nearby, so all the tall people, his progeny should get straight hair. That is nothing but the linkage. Okay. Then lethal genes. Some genes cause death. When they are present in the homozygous condition, they are called lethal genes. So here in the presence of lethal genes, the normal segregation, that is 3:1 ratio, is modified to 2:1 ratio. So these lethal genes were found in animals as well as plants. So in mice, allele for yellow coat color is dominant over gray. And when there was a mating done between yellow and gray, I got 1:1 ratio for the mice. That is, half of them are yellow color, half of them are gray mice. That means always the yellow mice are heterozygous, because yellow homozygotes are never born because there is lethality. Only, that is, the allele for yellow color is lethal, dangerous. If two copies of yellow color alleles are present, they will die. So only one yellow color allele and one gray color allele if present in heterozygous condition, they are born and they are yellow color in nature. Okay. So that's why only 2:1 ratio we can observe. Okay. The next is gene interactions. So where one pair of gene depends on the presence of a specific allele of another pair of gene. So that is called gene interaction. That means two genes, suppose here dominant allele is present, definitely that will interact with the dominant allele of other gene. Okay, like linkage, how it is. So such kind of interaction which will alter the phenotypic and genotypic ratios. Now all the ratios will be modified. So it is 9:7, 9:3:4, 12:3:1. Like that, if you do some examples, we can observe that there is a modification. Then pleiotropic gene effects. So Mendel always observed each character is governed by a single gene. But Nilsson L.A., he observed that some characters are controlled by several genes and each of such genes have additive effect on the expression. That means we have many genes for the same character. So all these genes, they will give more additive means if one gene is present, 10% expression. If 10 genes are present, 100% of expression. That means all they give synergistic additive effect. They'll give more expression. When number of genes are more, expression is also more. So this is called polygenic inheritance. And East, his name, scientist name, he demonstrated that polygenic characters are perfect in agreement with the Mendelian segregation. But later on, Fisher and Wright, they have given some mathematical basis for genetic interpretation of this polygenic characters. Then environmental effects. So genes can not only interact, not only like the expression and all, not only depends on genes, but even environment also they affect on this phenotype. Okay, so there is an interaction between the gene, genotype, and environment. And this importance of environment was first realized by Johansen. He coined the term genotype and phenotype. Okay. Then maternal effects. Mendel did not observe any difference between the direct and reciprocal process, but later there was difference in the reciprocal process, which led to the concept of cytoplasmic inheritance. So you know that mitochondria is always inherited from the mother, but not from the father. So the mitochondrial genes, mitochondrial related genes expressed in any individuals, they are from mother. Okay, like that means the maternal role is very important here. Got it? Then the next topic is linkage. So the alleles for the 7 pairs of characters studied in the Pisum sativum, they have homologous pairs of chromosomes, and he studied that law of independent assortment. And but later there was some deviation, and this type of coexistence of 2 or more genes on the same chromosome is known as linkage. So this was defined by Morgan in 1911 and he was awarded Nobel Prize in Medicine in 1933. And after some years, Bateson and Punnett observed in sweet pea that 2 pairs of alleles do not assort independently, and Morgan found the same phenomena in Drosophila and Hutchinson, and they actually explained, they given the concept of linkage. So linkage is the tendency of genes to be inherited in groups. So what are the key concepts here? Linkage involves 2 or more genes which are located on the same chromosome in the linear fashion. And they are involved either dominant genes or recessive genes or some dominant and some recessive genes. And these involve some genes which are closely located, and presence of linkage leads to high frequency of parental types. When linkage is present, recombination is not possible. So you can see they are more similar to the parents. Okay, but when there is a recombination, hybrids will be produced. And linkage may involve either two or more desirable traits, or all undesirable traits, or some desirable and some undesirable traits. And linkage is observed for both oligogenic traits and than polygenic traits. That is, few genes, when they are involved, linkage is formed. Linkage is possible. Then, besides pleiotropic, linkage is mostly found in the genetic correlation between various plant characters. And the strength of linkage depends on the distance between the linked genes. If the distance between the two genes is more, the strength will be less. Whereas if they are very nearby, the strength of linkage will be more. And if crossing over does not occur, all genes located on one chromosome are expected to inherit together. Thus, the maximum number of linkage groups in the organism is equal to the haploid number of chromosomes. Now we'll discuss about the phases of linkage. So there are two phases, coupling phase, repulsion phase. And they are given by Bateson and Punnett, but they could not give proper interpretation. But later on, Morgan in Drosophila, he has explained the phenomena of linkage, and he also explained the coupling and repulsion phases of the linkage. So what is this coupling phase and repulsion phase? So the linkage between two or more either dominant or recessive alleles is referred as coupling. Okay. So Hutchinson reported a good example in maize, where the genes for color of seeds. So color of seeds, there are two types of genes, colored and colorless. And shape of seeds, which are full and shrunken. So the colored seeds is governed by small c, and the full seed is given by capital S. Okay. So he made a cross. So here you can see, so the colored full seeds are capital C, capital C, capital S, capital S. So colorless shrunken seeds are small c, small c, small s, small s. When they crossed, so the F1 generation are all colored and full seeds. Okay. Then when F1 was test crossed, okay, with the double recessive parent, instead of 1 is to 1 is to 1 is to 1, they got some other ratio. Okay. So here you can see it is not 1 is to 1 is to 1 is to 1. You can see some are color are 4032. Then color shrunken, which are 149. Colorless are 152, and colorless shrunken are 4035. So they are not 1 is to 1 is to 1 is to 1, right? So different ratios they got. And this is because of the linkage. Instead of 50%, and the recombination was 3.6 instead of 50% in this case. And the linkage of dominant allele with that of the recessive allele is called as repulsion. Okay. So coupling means when they are both, they inherit together. Whereas the linkage of dominant allele with that of the recessive allele is known as repulsion. You can see, and Hutchinson observed this type of linkage when he crossed between the maize plants that have colored shrunken seeds and those which are colorless full seeds. Okay. So here you can see in the test progeny, again, the parental combinations were higher than expected when compared to recombination. So that means recombination was reduced than expected, but the parental combinations were more. So this is because of linkage. So as there is link, the genes are linked, there is no chance of recombination, and directly they got parental combinations only. Okay. So the next Haldane, he used the term cis and trans respectively for coupling and repulsion. Okay, then a name was given. Okay, so then the other topics I will discuss tomorrow, like types of linkage. I will explain linkage, crossing over. So because there are some calculations like how to do all these things are there. So we will discuss that topic tomorrow. Okay. Okay then. Thank you so much.